Welcome to Research Renaissance, presented by the Karen Toffler Charitable Trust. Embark on a journey into the forefront of cutting-edge brain science as we feature insightful discussions with a diverse array of guests, from early career researchers pioneering groundbreaking science to influential voices in investment communities, policymaking, and research institutions. Together, we delve into neurological diseases, their causes, treatments, and the quest for cures. Join us in advancing our understanding of the brain and addressing its ailments. Welcome to Research Renaissance. I'm Deborah Westphal, your host and guide through the fascinating future of the brain. Joining me today is Dr. Zhe Zhang, a research associate at Johns Hopkins. Zhe is also a 2021 Toffler Scholar. We will be discussing her focus on establishing a platform to find genetic modifiers that can improve cell survival and slow the progression of ALS. So welcome, Zhe.
Hi, Deb. Thank you for inviting me.
I'm very excited for you to be on, and congratulations with your recent promotion. We just kind of talked about that. That's wonderful. So let's just kind of start with what sparked your interest in neurodegenerative diseases, particularly ALS. And can you talk about, your medical training and how you became a researcher?
All right. I think that's kind of an interesting story because I always dreamed about being a doctor since I was 4 years old. At that point, I always dreamed about being a surgeon. Never thought about doing research at that point. Well, and then I made it into medical school. And while I was doing rotation in the 4th year of my medical school, I actually did that rotation in the Department of General Surgery, where I had this patient who suffered from Alzheimer's disease. So he came in because of the bed sore, and he and his wife actually lived in the hospital for many, many years. And his wife always talking to us while we doing the treatment, talking about their story, their love story. I thought that's lovely. I always enjoy those stories, but when I suddenly look back to him, I saw he has no expression in his eye. He cannot move. He cannot feel that anymore. That makes me really sad. And that was the first time I was kind of asking the question, what I can do for the patients of neurodegenerative disease. And then I think there are many opportunities that I encountered the patients with neurodegenerative disease in this department. And then I kind of have my interest shift a little bit towards to the research. I'm asking more why questions instead of like protocols, thinking about how to make the diagnosis and treat the patient. I think that's my first spark for the research in this field.
That is it's a sad, but it's a beautiful story. And I can see why you became, kind of touched at that, at that moment. It's hard though, right? Going from being a doctor to research because, and correct me if I'm wrong, in the clinic, when you're with patients, that's kind of your day job. And then at nighttime, you go to the lab and you do your research. Is that correct?
I think I'm kind of lucky because I haven't really get to the very hardcore clinical practice yet at that point. I finished my bachelor degree with clinical major, but then I switched completely from the clinical part to the basic research part, where I did my master in embryology and histology. And then I pursue my PhD in Australia. So I don't really have the experience of doing 2 jobs at the same time, doing clinic and basic research at the same time. But I think I do met many challenges throughout the way. Because when I first make the transition, I noticed there's huge difference between these 2 jobs in terms of the mindsets, right? So as a doctor, you think more about they're patient-centered. Then you have to think about the treatment you want to see into immediate outcome. But as a researcher, you don't know. you have to spend many, many years working on the same project till you can really find something or nothing in the end. I think that's kind of different mindsets that you have to have for these jobs.
I imagine that the mindset fits you well. This more future focus, more kind of search for the answer, understand the problem rather than this urgency to meet the need of the patient immediately. Is that kind of correct?
Yeah, I think that's the major difference between these two jobs. But as a doctor, you always want to see a good outcome for the patient. But as a researcher, you always want to know why. I think that's kind of the thing I had at the very beginning. I always pay more attention to the outcome. At that point, I don't really care about mechanism for that much. At the very beginning of my research, I probably would think, well, this drug works, let's put it in mice and see whether it works. But now, the more research I do, I more pay more attention to the real mechanism part because good mechanism study can also boost your translational research. I kind of get that point now. Yeah, so that's the biggest difference between these two.
So why ALS? And did it have something to do with your transition when you went to Australia and went to university there?
Right. That's kind of a good story as well because I was recruited to Dr. Simmons' lab doing sulfate metabolism in the developmental disease at that point. But then we study lots of sulfate transporters. We study lots of sulfate metabolism related structures, such as extracellular matrix. But at that point, we have a collaborator who is a kind of pioneer established in Australia that he studied the SOD1 mutant in ALS. So they have these animal models. We're trying to help in describing some structures wrapped around the neurons that could contribute to the impairment of the neuronal function. So that was my first study related to ALS.
And then you talked about brain development. Is this when you were studying, just to clarify, was it more in the development of, from adolescence, from babies to adolescence and that brain development?
Yeah, so we're kind of trying to understand why you have issue with sulfate metabolism, why you start to have some autistic-like phenotypes in the mouse. It's not actually in human, but we were thinking maybe that's kind of you can translate that to human disease. So we were just curious about how this autistic-like behavior happened in a mouse model. But then, yeah, we also working on ALS later on. So yeah, that was the connection. That was the first set of experiment I did. And also at that point, it was the ice bucket challenge. It's around that time. So it's suddenly this ALS, this very rare disease exposed to general public. So that also interests me a lot. And also, I went to a neuroscience society annual conference. So I met many people there who study ALS. I think it's like many reasons why I'm here.
Yeah, and that ice, and I forget who started it, but it was, dumping ice on big, coolers of ice water and taking the challenge, and it kind of went viral on social media, and it just kind of highlighted the term ALS.
Right. So it's 10 years now. I think
Mhm.
they're celebrating their 10th years of Ice Bucket Challenge, which I think it really promote ALS to let general public know the term. And I think it helps with the diagnose, the early recognition of the disease. It helps a lot.
Is that kind of in maybe we study some of these diseases as aging diseases, but many of you researchers also question how do they develop, especially in adolescence or in early childhood. Are there other kind of common, you mentioned that connection or that appearance that it looked like autism. Are there other kind of childhood diseases or conditions that, may somewhere along the way in the future, we go, okay, these are a part of this too, whether that be epilepsy or, whatever they may be.
Yeah, I personally think it's possible because it's in the genome. Like, for example, for the type of ALS I studied, that the C9 mutation is always there, but you only see it while you're aging. So it probably does something at a very early on, but we don't know. So it is an interesting aspect of the disease study, I think.
So, I mean, you mentioned the type of ALS that you study. How many types are there? And can you talk more in detail about what your research focuses on?
Yeah, so for the ALS, there are two general populations, sporadic ALS, familial ALS. So we're studying a type of familial ALS, which is caused by C9orf72 gene mutation. So this one contributes to more than 30% of the familial ALS. Of course, there are also many other mutations, but probably each one only takes a relatively smaller proportion. So yeah, as I said, it's contribute to more than 30%. So that's why we're like super interested in. And for our study, our lab is really interested in this type of ALS because when you have the C9 mutations, you have repeat expansion within this gene. So these expanded repeats can produce toxic proteins. So people have shown that these toxic proteins are leading to cell death. That could partially contribute to the neurodegeneration. So in our lab, we're particularly interested in developing platforms that we could find those genetic modifiers that can decrease the production of these toxic proteins, as well as maybe you cannot decrease the protein level, but you can improve the cell survival without touching the production of protein so that you make your cells more resistant to these toxic proteins. That's what we're trying to do.
So, what does the toxic protein do? Like, does it kill parts of the cell? Does it gunk up the mechanism of the brain? What does it do?
I think it's pretty broad impact. At the very beginning, you have this protein produced, so it might interrupt the function of different organelles, such as like mitochondria, ER, and all those kind of important machinery in the cell. But when you have more, so they become aggregates. They cannot dumped out of the cell, and then these aggregates can, you can say, is attracting other proteins around it. And maybe that will lead to the impairment of the protein function. So there are many mechanisms, I think, currently people studying, like why these peptides, these proteins are doing harm to the neurons. And also, well, it might not directly leading, one part is directly leading to cell death. Another part is with these proteins, it might also interrupt other cell types that in the brain. It's not necessarily neurons. It could be astrocytes, microglia. Those cells, when they're cannot functioning properly, they also do harm to neurons. So I think that's different layers of mechanisms, like why these toxic proteins are killing cells. But in the end, you see the death of neurons as the outcome. And then people lose function. That's the process.
So when you say you're trying to develop a platform to understand these modifiers and possibly therapeutics to address the disease, what is that? The platform, is that a what is that? Is that a taxonomy? Is that an understanding? What is the platform?
Yeah, that's a very good question. I think initially when we first built up this, we just want to put it in a simple way. It's genetic screening, but we want to know if there's any existing drugs, for example, that can help the survival of neurons. So we do the screening in a way that we knock down some of the genes that they have inhibitors on the market. Is FDA approved or it's in a clinical trial. We have a pool of that. So we knock down those genes and see which ones, by sequencing, we know which one can improve cell survival. We do have a pretty successful experience with this. The most recent paper we just submitted is actually we found one that when you're knocking it down, it can significantly improve cell survival. But I think it's also later on, we think it should be beyond that. We want to know the mechanism, like in general, when you're regulating different pathways, what will you see? Which is the most significant pathway in this process? That's the question we want to ask. So we also perform this screening in a genome-wide scale. So we knock down like more than 20,000 genes and see which ones can improve the survival. We're working on that part now. But at least for the first part, we know there will be something you can screen out and it could be beneficial for different models.
And are you using the mouse models to do the screening, or are you using from samples from the brain bank, or are you using actual cells?
Right. So I think thanks to the genius researchers, they engineered human iPSCs. Like they put in the kind of, you can say it's a control, like controlling the differentiation process. So basically we got an iPSC and we turn it on and it start to differentiate to cortical neurons. So we actually perform the screening in human neurons.
And where do you get those human neurons?
Yeah. So there are some established lines out there that people engineered based on that. It's not only like differentiating to neurons, it can also differentiating to other type of cells, but they're all coming from a pretty standard human iPSC line. I think with that makes your study robust, right? Like when we start the differentiation, we could have thousands of neurons from the same resource. So it's kind of well controlled. As a good quality control, it's well controlled. But of course, like after we screen out something, we always validate it in different platforms. It's either in these cells or it's other patient-derived neurons. I should have a big shout out to Julia from BU. She's the expert in this field. She's done so many wonderful things using these models. That's also something we thought very helpful with our research. And also we validate, if we see some protein change or RNA change, we always go back to the patient postmortem tissue. And I would like to say big thank you to those families who donate postmortem tissue. That I think that truly promote our research. I think they're a very important part of this process. I truly thankful for that, appreciate what they've done. Yeah, so that's kind of the models we use. We start from very basic, and then we move towards to really disease-related models.
Yeah, it's fascinating because I actually had Julia, Dr. Julia TCW, on the podcast, and she talked about how you can take, the science now is you can take a scratch of an individual's cells off their arm or somewhere and grow what she called grow a brain in a petri dish. And
yes.
And that science, that stem cell, I remember many decades ago, just even talking about stem cells were very controversial because, where they were going to come from and, some of the ethical parts of that. But now using just a scratch of somebody's, a volunteer that is donating that and being able to grow the cells that you need to do your testing, that kind of stem cell research is really amazing, and it's allowing us to do some incredible things.
Yeah, it really boosts the research because once you have that line, basically you have the cells from the patient that contains their genomic information. So it's like mimicking what is happening in vivo. Yeah, that's terrific. I was like mind blowing when I first started to do this kind of research. It was so cool.
Yeah, it's fascinating. It's just absolutely fascinating. And like you said, the people that come in and donate, especially if they're being diagnosed it early on, may allow for maybe a deeper understanding. So, how do you envision your research on these genetic modifiers leading to therapeutics? And this is probably the time to ask you about the CRISPR technology. And I don't know if this is a part of your research or a part of that future that you envision, but can you expand?
Yes. So basically, I think when we talk about CRISPR, I personally, according to my experience, I would like to separate it into two parts. First one is the platform I just talked about. So CRISPR I or CRISPR-A in terms of manipulating gene expression level. So those platforms are good for the screening for modifiers. And I want to tell you a story that we screened this hit out, we validated it in different models. And now we're really seeking opportunity to make it more like a product or translational approach. We're trying different ways of knocking down this gene by siRNA, different kind of method, including antisense oligonucleotide technique. We're trying to do all these different manipulations and see if it could also improve cell survival or neuron function in a more in vivo model. So currently all the models we have are in vitro. So it's in the cultured cells. Yeah, so we haven't get to the point of testing it in an animal model, which harbors this C9 mutation. So we're trying to do that now. So I think that's kind of how we translate it from this basic screening to the potential pharmaceutical approaches. And another part is CRISPR. There are also many methods designed using CRISPR as a way of doing gene editing. That's also something we're working on. I think there are lots of companies do that already, like in the central nervous system, they manipulate the key mutations, they correct them. So that's more like from the root. You solve the issue from that. So you have the mutation, you correct the mutation so that you don't have all those pathological changes anymore. I think that's another thing that CRISPR technique can help.
Yeah, that definition of CRISPR, or that the second one is kind of to me, it's kind of you're rebooting your cells to just kind of start all over and not have the disease. Is that correct?
It's kind of, I think it's kind of hard for neuron to boost it like completely because they're not proliferating anymore. I think it's more like go in there and correct the existing ones. But I think for many diseases where the cells proliferating fast regularly, it is kind of boosting.
Yeah, do we know why the neurons in the brain, we have the number we have and that's it?
Yeah, so I think, yeah, it's kind of interesting, right? Like, I know people know we have neurogenesis, but it looks like very small proportions. So that's critical for the formation of the maybe new memories or something. But yeah, overall, we only have very small proportion of the neurons that can generate it after adulthood. That makes neurodegenerative disease like very hard to work on. Like Alzheimer's disease, we spent so many years using so many different therapeutic approaches, and we haven't get to the point of correcting them. I think the same case with ALS.
Yeah.
Yeah, I think that's due to the feature of central nervous system.
Yeah, I guess I never thought about that. But if our cells were always dying and replenishing, what would that do to our memories? What would that do to, the things that we house in our head if it's always changing? I guess, I don't know, maybe it's a simplistic way of thinking about it, but it's kind of.
I asked that question a lot while I was doing my PhD because at that point I figured sulfate metabolism is important for neurogenesis in the rodents at least.
Yeah, that's super interesting. And you mentioned on this the kind of this first use of CRISPR and how you're doing it for gene modifiers to, create the platform to be able to test the therapeutics or test. And then you said, you're kind of focused on a product. To me, my background would say, oh, that's interesting. You're going to try to commercialize this somehow. Am I off track there?
I think you're right on track. That's what we're trying to do. But also, we realize it's not something that we can easily do. As researchers, we don't really have that kind of background in terms of commercializing. I think that's kind of the point. We need collaborators from farms and all those people from biotech that we put force together and make this happen. At least currently, we do have some people approaching, so that hopefully that will be something that we can do in the near future.
Yeah, because there's investors. There's investors that are very interested in helping those academic entrepreneurs with that. There's also, kind of organizations like Tamir Foundation that can help. Once you've kind of established, like, okay, this is the product. Now I got to figure this out. That those first time CEOs, but that is a challenge taking it from the lab to the market.
Yeah, I think also before that, there's also several steps before we can actually put it on the market. It's like this gene we know from the screening, but then we don't know like globally when you knock it down or inhibit its activity, what the influence will have, like in general as a human. So I think there are still lots of things we need to test before we go even further, and we have to be always super cautious about it. So I think as researchers on our side, we still have lots to do.
Yeah. When you say globally, there's challenges, are you thinking about that from a geographic globally, that the disease is kind of different in different parts of the world? Or are you thinking globally as far as lots of challenges and problems?
I think globally, usually, like to me, it means this human, like the actual person, like human being. Because when we study molecular mechanisms, like we really dive into RNA, DNA, like molecule level. So when we say like globally, it usually means like real human beings. Like we ask the patient to take orally take medication. So that kind of impact that usually referred to that part.
Yeah, that's fair because we're all very different. We have different genetics, different life journeys, different, just everything.
Yeah, especially when it comes to the patient, there are so many people who study like biomarkers. Like if you see those plots, like the distribution of the data is like huge. The variation is huge. I think that's also kind of the challenging, or like when you do the real patient clinical studies that you have to face. It's not like culture in a petri dish. We have everything well controlled. Every cell have similar background and similar treatment. Like human patient is different level of variation. I think in terms of that, I think that's also make the clinical study super critical for the all this therapeutic method development.
Yeah, I think we're going to find, in the future that just even different environmentals, economics or, access to food, access to clean water, how much pollution we were, had, exposed to. I think that's going to really create issues for those clinical as we try to get very precision on that patient.
Yeah, so that's why I feel like why global studies like Researchers United and all the patients or doctors work on something together. I think that's kind of highlighting the global collaboration, how important it is. Then you have maybe bigger picture of the disease. It probably helps you understand the disease mechanism.
So we talked about some of the technological breakthroughs that have kind of happened in the last 10 years with, stem cells and CRISPR and just even, 20 years ago with the whole DNA coding, decoding of the DNA. What kind of emerging technology or emerging tools do you see that are going to really kind of help your research, or maybe already are helping your research?
Yeah, I think that's kind of the point I want to make. Is like we use iPSCs heavily, right, routinely, and we have all this CRISPR based screening platform, and we use some pretty fascinating imaging method as well to really give you the resolution of single RNA level. So it helps you to understand the disease mechanism for sure. But I personally also thinking about maybe something like related to functional MRI or something like that could also help to understanding the disease, because those are the imaging or the exams people usually will do, like when they first notice something is wrong. So maybe you track them throughout the time, you can find something. But I think it really depends on the questions that we want to ask.
And that functional MRI, that's, if I understand what you're saying, is that's kind of looking at the disease manifestation over a time frame. So what's changing? Where is it changing? Trying to understand why is it changing?
Right. I think that's the idea.
Yeah.
I think probably better to have some invasive method, because you can't do biopsy all the time to the patient. There better to be something that you can easily get, can easily track throughout the disease progression process. I think that's also highlight the importance of biobanks, because you can drain blood or you take CSF for different time points, then you see the change of certain biomarkers. So I think that would be very helpful as well.
Do you work with others or collaborate with others, or just even in your network stay in tune with some of these emerging evasive techniques? There's things that I've seen where either electromagnetic maybe hat, or some sort of thing, cap on your head, or even more evasive, kind of drilling little holes or putting little probes in your head. Is there any of that kind of collaboration?
No, we don't have that kind of collaboration for now, but I think it will be very interesting to have. Like, for example, for the ALS patient or many other neurodegenerative disease patients, like before the real disease onset, like you can see severe symptoms before that stage, you can detect the hyperactivity of the brain. I think that's kind of can be done by those equipments you're just talking about. I think that's kind of maybe if it is well controlled or developed, maybe that could be a way of predicting disease, or um, that would be very important. It can consider that as different kinds of biomarker.
What's the overlap from what we understand today? In your opinion, what's the overlap of ALS and maybe dementia or Alzheimer's? And the reason why I'm asking that question is not too long ago, there was a lot of misdiagnosis of, people saying they had Parkinson when they really had Lewy body dementia. What's kind of what we know today, that overlap?
I think genetically, ALS, FTD, they have very clear genetic overlaps. Just like, I think that's a very good question. C9, this C9 mutation, it is well known. It's an overlap between FTD and ALS. And in terms of pathology, maybe there are also some like shared feature. Like for example, TDP43, you can consider that is a protein that people usually found it can aggregate, aggregate it in the neurons. So that pathology can be found in it can be found in more than 90% of the ALS and more than 50% of the AD. So definitely for those diseases, they share many genetic and also pathological features. Sometimes even I ask myself whether you can consider them as a whole general neurodegenerative disease.
Yeah, which makes it interesting going back to the brain development, because Is there something happening in the development of the brain that is more kind of generalized, that becomes more specific as we get older? I just it's kind of curious.
Yeah, me too. I think that's an interesting aspect, but unfortunately, I don't really have the expertise in studying that for now. But I will keep my open for that.
Well, that's why it's so important. I mean, there's so many of you looking at these diseases in very different perspectives because it's very complicated. The brain is very complicated, and it takes all these perspectives to kind of, you know, get that understanding. I'm imagining that you do collaborate though with other researchers. I mean, you mentioned Julia TCW from BU, but how important is it to collaborate with others to boost your understanding?
Yeah, I think it could be of different levels as well. So, in terms of, like, for example, Julia and also Dr. Michael Ward from NIH. So, he's the one like making this platform for all these engineered iPSCs. So with that kind of collaboration, we get all these tools that we can work on. And also like when we collaborate with the biobanks, we get the patient specimens so that we can examine our hypothesis, like say if we have some protein up or down, if we think that's very important thing in terms of predicting the disease progression, then we examine it using those samples, and we see if it fits with our hypothesis. Maybe it stopped there. Maybe it's still a hypothesis, but maybe you find a clear link between this. And also, I would say another layer of collaboration is more like maybe some researchers, we have similar interests, but we have different technologies. So we can use different technologies to tackle the same question. So by applying these approaches, we can really boost our understanding. It's just like the collaboration between our lab and also imaging lab. So we can make some reporters. We can see where the RNA got exported into into the cells, and whether they can translate it into these toxic proteins. I think all these kind of collaborations on different layers, it can really help our research. I think that's super important. And also collaboration between biotech farms that really is kind of on different layers of promoting the translational research.
Yeah. Yeah. So when you look out, 5, 10, 15 years, pick your time frame, what's your greatest hope for, your research and maybe the impact it made, or the community researching ALS? What is your hope?
I would put it in a 10 years frame or even longer than that, because I know it's super difficult to study neurodegenerative disease. But I think everyone in this field, we're aiming for therapy. And in terms of the research, I hope people can be more open to their studies, like more collaborative. I think really that something can help to develop therapy for the patient, because after they diagnosed for ALS, we know it's usually 5 to 10 years, 3 to 5 years. They don't have much time. So we have to hurry up.
So when you say, you wish that people would be more open to do the studies, what is that? What's the concern there? Is it support and money to do the studies? What is the concern about being more open to do the studies?
I think first part, funding, it is a factor. And also I think we have like, for example, we have really good hits from the screening, but how to move forward. So we need biotech farms to come in and help us. So when they make decisions, maybe they can tolerate more. Like they don't, maybe if they don't need a like very full package, maybe they can they can just try it with the solid data we already have. Maybe that can already boost the research. I think that's kind of the thing, like people commercializing. Of course, I understand. It has risk, but sometimes it's a balance of risk and the benefit. Like maybe people can find a better way to balance it.
Yeah, I think you're describing just the complexity of commercializing, because it's very different camps, right? A researcher is different than kind of the funder, which is different than a more mature pharma company that has the ability, and they all speak different languages. They all have a different ecosystem. That network, and does Johns Hopkins have a pretty robust kind of program to help researchers, or is it, in this area, or do you have to have your research pretty set before you can use those kind of, entrepreneurial, processes, I guess, within the university to help you find or connect to pharma companies?
I think that's doable. At Hopkins, I know they have an organization called JHTV. They can help you file patent. They can connect you with farm companies. They're pretty open to the collaboration. I think that's kind of a good attitude to have in this field, like trying to promote basic research, promote it to the translational part. I think that's good effort.
Yeah, I think there's, some of the bigger universities have that as, but they're still even hard, right? You're doing your day-to-day thing with a lot of hours, and learning another kind of language, the language of business can be very difficult.
Yeah, so I think it's important to have such organization. They can help us translate our language because we're doing basic research. We don't really know much about business. So we need someone to help us doing this work, like translate what we want to say and can express it clearly to the farm, to the companies.
Yeah, yeah. So what would you say to those kind of people that are behind you, that are thinking about medical school, thinking about research, looking at what they want to do, for their career? Any kind of words of wisdom to kind of help them think through those decisions?
I think considering myself is still pretty junior. I would like to say something to myself and also even junior ones. I think for us, it's important to stay curious and stay passionate about what we're studying at and be cautious because whatever we're studying, it might translate to the real therapy. You have to be cautious about it. I know there will be days up and down. Most of the days, I would say, is down. Nothing works. It's just a bad day in the lab. But there will be days that are happy, like patient contact you, ask you for clinical trials or those kind of things. I think those days are super rewarding to me. I think regardless, we have to keep in face that we're fighting together to this disease. We're trying to help patients. We will figure out something that can help to improve their life, to get more time for them with their families. I think we have to keep that faith.
Yeah. Yeah, it's really interesting too to consider you. You said you're a junior person, but the amount of schooling that you've gone through and the amount of learning that you've gone through. I think people, it's the career of a researcher is very different than I think a lot of. It's sometimes it reminds me of an apprenticeship where you have to be there for 15, 20 years before you can take lead on something, and it's quite extensive.
Yeah, it's pretty long journey, I have to say.
Yeah.
But so far so good. I'm still enjoying my study. I still like the way I perform every day in the lab. I still have hope. So that's good.
Yeah, and it sounds like you're still very passionate about that human on the end of this of, solving their problem and hopefully making them healthy.
Yeah. I wish one day we can develop something that can reverse this disease at all.
Yeah.
I have that hope.
That probably drives you too on those down days that you talked about. So, yeah. So as we kind of wrap up here, Z, is there a way if people are interested in just finding out more, what you do, collaborating with you, maybe helping the translation of your research, or just finding out more, how would they get in contact with you?
Yeah, so if people are interested in my research or any suggestions from me or any help, they can Google my name at Johns Hopkins. So I'm pretty sure they will find me. If it's really like the translational collaboration, those kind of things, can also reach out to my PI, Shu Sung at Johns Hopkins at the Department of Physiology. We're always here. So yeah, you're welcome to contact us and discuss about anything you're interested in.
That's wonderful. I really appreciate your time. I know that you had a big experiment that you were trying to wrap up as you tried to make this call. So I really appreciate that, and it's been wonderful to catch up with what you're doing.
Thank you for having me.
Thank you for joining Research Renaissance. For more information and to stay updated, subscribe for more episodes and visit our website at tofflertrust.org. Feel free to call us if you have any thoughts, suggestions, or just want to find out more about the Karen Toffler Charitable Trust. Until we meet again, onward and upward.